Hi there, my name is Mariah Angel Cuyson, and I'm currently a second-year master's student majoring in cell and molecular biology under Dr. Andrea Sway's lab at San Francisco State University, where I also completed my bachelor's degree in microbiology and Asian American studies back in 2022. And so Lyme disease is the leading tick-borne disease in the United States, and it poses a major public health threat to both humans and wildlife. And so in the western United States, it is caused by the pathogenic spirochete Borrelia and transmitted by the tick species Ixodes pacificus. The tick life cycle contains 4 life stages: egg, larva, nymph, and adult, transitioning into each life stage upon taking a full blood meal. Eggs are hatched uninfected into larvae and acquire Borrelia by attaching and feeding on an infected reservoir host such as a squirrel or a mouse. Engorged larvae will then detach from the reservoir host, molting into the next life stage as nymphs, and proceed through the remaining life stages of their life cycle. Ixodes pacificus feeds on a variety of rodent and lizard species. However, western gray squirrels, wood rats, and deer mice serve as the main reservoir hosts of Lyme disease in California, and this is because they harbor Borrelia and transmit the pathogen to the tick upon feeding. And so my research project investigates the impact of host blood meal identity on tick microbiome composition by analyzing a variety of non-competent and competent reservoir hosts of Lyme disease. Specifically, I'll be looking into the microbial composition of ticks that have fed on different species of rodents and lizards which vary on host competence, and this is the ability for a host to harbor and transmit the pathogen to another host or vector. And so the new information I hope my study will bring to the field is really understanding how the microbial composition for each of these hosts will compare, along with the relationship among host competency and microbiome composition and species richness. And so to do this, I'll be performing next-generation sequencing on the hypervariable V3 to V4 regions of the 16S ribosomal RNA gene, and this gene is widely used for the phylogenetic and taxonomic classification of different microbial communities, including bacteria, fungi, archaea, and viruses. And so here's the overall microbiome sequencing workflow, which consists of library preparation and quantification, performing a sequence run using the Illumina MySeq system, and using quantitative insights into Microbial Ecology 2, or CHIME 2, which is a next-generation microbiome bioinformatics platform used to analyze the microbial community composition and richness for each sample. And so lastly, I want to say a massive thank you to the ARCS Foundation Northern California chapter, as this scholarship has really allowed me to grow as a master's student and continue to pursue my passion for scientific research and public health. Thank you.